Hey everyone, and welcome to this explainer. Today, we're doing something really cool. We're explaining the chapter on bioprocessing and biomanufacturing. And honestly, I'm just absolutely thrilled to take you on a bit of a factory tour today. But okay, we're not talking about a factory that builds cars or smartphones. No, we're looking at how humanity has figured out how to harness microscopic life. Think bacteria, fungi, plant cells. We use these tiny things to mass produce essential biochemicals that literally save and sustain our lives every single day. So here's our roadmap for the chapter. We're going to start with the historical penicillin scaling problem, then we'll examine the anatomy and the different types of bioreactors, and finally, we'll actually walk the biological factory floor by exploring upstream and downstream processing. All right, let's jump right into section 1, the penicillin scaling problem, from petri dish to mass production.
You know, to really understand why bioprocessing is just so absolutely crucial today, we kind of have to look back at the massive historical stakes that essentially forced this entire field into existence in the first place. It's that classic happy accident story. Picture this. It's 1928, and Alexander Fleming leaves some petri dishes unwashed at a London hospital. When he comes back, he notices that the Staphylococcus bacteria he was trying to grow couldn't survive near this random invading mold. Well, that mold was Penicillium notatum, and it literally changed medicine forever.
What? Here's the massive challenge with that. Discovering a wonder drug is completely useless to the general public if you can't actually get it to them, right? The natural quantities of penicillin produced by this mold were just incredibly tiny. And during World War II, there was this desperate, urgent need to treat wounded soldiers with this brand new antibiotic. So the scientific community faced a monumental scaling problem. They had to figure out, how do we take a fragile, living mold and somehow force it to produce its wonder drug on an industrial scale? Well, that urgent need right there, that's what gave birth to the modern field of biomanufacturing. But before they could even think about building the machines, scientists really had to understand what they were actually trying to harvest. And that is secondary metabolites. See, while cells make primary metabolites just to survive, secondary metabolites, things like defensive antibiotics, dyes, or enzymes, well, those are extras. The microbe doesn't strictly need them to live, but for us humans, they are incredibly valuable. So the ultimate goal became all about coaxing these cells into pumping out massive, massive quantities of these specific secondary metabolites. Moving right along to section 2, anatomy of a bioreactor, building the microbe's home. Okay, so to actually coax out those secondary metabolites, we need an unbelievable level of control. Enter the bioreactor. It's essentially a giant high-tech vat. Think of the agitator shaft right in the center, like a spoon stirring your morning coffee. It keeps all the cells perfectly mixed up. Then you've got the sparger at the bottom. That acts kind of like the lungs of the operation, pumping in sterile oxygen so the whole culture can breathe. The baffles on the sides, they break up the water vortex so the mixing stays nice and smooth. And finally, the outer jacket serves as the ultimate climate control system. It circulates water to maintain the exact, perfect temperature that these microbes need. So the absolutely crucial point here is that a bioreactor has to be the perfect home for microscopic cells. First off, it must remain absolutely sterile. You do not want rogue bacteria crashing the party. It also needs to provide a steady air supply and uniform mixing that won't physically rip the fragile cells apart, something we call shear stress. Plus, it has to constantly monitor and adjust things like temperature and pH, because at the end of the day, if the cells are perfectly happy in their environment, they're going to multiply quickly and become highly, highly productive.
Which brings us to section 3. 3 types of bioreactors: stirred, airlift, and bubble. Now, because different cells require different environments, not all biological factory floors are going to look exactly the same. We've got a few main designs. There's the traditional stirred tank, which uses a mechanical impeller, basically spinning blades to mix the broth. Then there's the airlift reactor. This one relies on a central draft tube, using air currents to physically lift the fluid and the cells up and down. And finally, the bubble column, which uses low-shear sparger jets. And that last one is actually incredibly important for highly sensitive animal or plant cells. Why? Because those delicate cells would just be totally destroyed by the spinning blades of a traditional stirred tank.
Let's dive into section 4, upstream processing, setting up the factory. Just to make sure we're all oriented on our factory tour here, every single bioprocess is split into 2 very distinct halves. Upstream processing is all about preparation. It's getting the food ready, sterilizing the equipment, and actually growing the living materials. Downstream processing, on the other hand, well, that's the cleanup phase. That involves extracting the valuable product from the biological soup,
purifying it, and safely getting rid of the waste. But let's take a closer look at the upstream side first.
So, upstream processing is broken down into 4 incredibly precise steps. First things first, you have to design the perfect artificial food, known as media, for your specific cell type. Second, you absolutely must sterilize everything. Seriously, one single rogue contaminant can ruin an entire 10,000-liter batch, which is a total nightmare. Third, you produce a healthy starter batch of cells, and we call that the inoculum. And fourth, you dial in those environmental conditions. The pH, the temperature, the oxygen levels, you get everything just right so those cells can really thrive.
But you might be wondering, how do scientists know exactly what to feed these cells? Well, it all comes down to math. There's a stoichiometry equation that acts essentially like a recipe. It maps out exactly how carbon and nitrogen are converted into cell biomass, plus your desired product, and of course byproducts like carbon dioxide, water, and heat. By calculating the exact cellular yield coefficient, biomanufacturers can design highly economical media. It minimizes wasted ingredients and, most importantly, it maximizes that final product yield. It's fascinating to trace the journey of our tiny worker cells. They start out super small, just on a little solid media plate. Then they're moved into a liquid suspension culture inside a shake flask so they can start to multiply. From there, it's time to scale up. They move to a pilot bioreactor and then finally into those massive industrial-scale tanks that can hold thousands and thousands of liters. It is a very carefully, beautifully orchestrated expansion. But here's the catch. That massive industrial tank, completely useless if you don't start with the absolute perfect inoculum, or starter batch. This is literally the make or break element of upstream processing. The cells must be in what's called their exponential growth phase. That just means they're actively, rapidly dividing. They need to adapt super quickly to their new environment, have a large enough starting volume, and remain absolutely 100% free of contamination. And crucially, they must not have mutated, because if they mutate, they might lose the capability to form the exact biochemical product we actually want.
All right, moving into the home stretch with Section 5, downstream processing, the final harvest.
So there's a critical pivot point that happens in this whole process. Once the cells in the bioreactor have done their job and produced the absolute maximum amount of our secondary metabolite, the upstream process officially ends. That's when we transition right into downstream recovery and purification. Downstream processing is basically a highly meticulous separation phase. Step 1 is all about extraction, recovery, and the strict purification of the final product. Whether we're talking about a pharmaceutical drug, an industrial enzyme, or even a food additive, it has to be perfectly separated from the cell mass and all that leftover broth. But step 2 is honestly just as critical, and that's the safe, responsible disposal of the process effluents. You can't just take thousands of liters of biologically active waste and dump it down the drain, right? It has to be treated and managed properly.
Wow, we've come a really long way from an unwashed petri dish in 1928, haven't we? By mastering the strict control of these microscopic environments, bioprocessing basically allows us to scale up nature's most complex chemical factories. So I want to leave you with this thought to chew on. If we figured out how to harness a simple mold to mass-produce antibiotics that literally change the course of a world war, what massive global challenge is biomanufacturing going to solve next? Could it be lab-grown food to feed the world? Or maybe revolutionary new biofuels? The possibilities really are endless. Thanks so much for joining me on this explainer today, and as always, keep exploring.